AS Level Biology 9700, and this is the second topic and the third video on this topic. So the today's subtopic which we are going to discuss is the starch. And starch, it is a polysaccharide, and polysaccharide which is made up of glucose molecules. So the basic formula of starch is very similar to that of glucose. It is very similar to that of glucose because it is made up of glucose molecules which are joined together with the help of glycosidic bonds, and there are two forms of starch. And one form has no branch. It is in the form of long chain, which is called as amylose. So amylose is made up of more than 25 glucose units per one molecule of amylose. So it means, so glucose molecules, they are joined together and then they form the starch molecule, and starch molecule the form which is amylose. So if we discuss this amylose, then you can come to know that amylose is formed by the condensation reaction. The condensation reaction takes place. Condensation means in which the water molecule is removed. So here in this case, you know that there are two types of glucose molecules, alpha glucose and beta glucose. So the amylose is formed, or starch is formed by the alpha glucose molecules in which hydroxyl group is in the downward direction. So in this way, a long unbranching chain of several thousand 1,4 linked glucose molecules will build up. That is, the first carbon of one glucose molecule make a glycosidic bond with the carbon number 4 of the second molecule. So in this way, a long chain of glucose molecules is formed. So that chain, which can be more than 250 glucose molecules, so that is called as amylose. So these chains, you can say when there is formation of 1,4 glycosidic bond, the chain coil up into helical structure like spring. So in this way, a compact molecule of amylose is formed. So I want to show you in the next slide. You can see the formation of amylose takes place in this way. That is, there is one glucose molecule, carbon number 1, and it will make a bond with the carbon number 4 of the second molecule. So then carbon number 4 with the carbon number 1 with the carbon number 4 again. So in this way, there is formation of alpha 1,4 glycosidic bonds. Because in alpha glucose molecules, the hydroxyl group is in the downward direction. So here the formation takes place and glycosidic bond by the process of condensation reaction. So condensation reaction in which in every joining of two glucose molecules, there will be the removal of one water molecule. So in this way, a long chain of the starch takes place, and which is called as amylase. It is non-branching form of the starch molecule. And when a long chain is formed, that long chain make this helical structure. You can see, so this helical structure, many glucose molecules are joined together with one another by making glycosidic 1,4 alpha 1,4 glycosidic bond. And the next form is amylopectin, which is a branched form of starch. So there is two types of bonds which takes place, which are formed in the amylopectin. The 1,4 glycosidic bond in which I have told you that the glucose, one glucose will join with the other glucose molecule, carbon number 1 of one glucose molecule and carbon number 4 of second molecule. So in this way, 1,4 linked alpha glucose molecules will make amylose, and the same will take place in the amylopectin. But this is a branch structure. So there is another there, as for example, so this chain is 1,4 linked alpha glucose. So there will be a branch structure. So in this way, so it will be the formation of this branch structure. There will be 1,6 linkage. 1,6 linkage means glucose number 1 will come join with carbon number 1 and the glucose 2 will join with the carbon number 6. So in this way, a branch structure will be formed. So here you can see in this slide the branch structure of amylopectin. So here you can see this chain is making 1,4 glycosidic bonds. The whole chain has 1,4 glycosidic bond sequence. Now you can see a branch is arising from, this is the carbon number 6. So a branch is arising from here. So you can see this is the carbon number 1, C1, and this is C6. So there is formation of this bond, of this glycosidic bond between carbon number 6 and carbon number 1. So this is 1,6 glycosidic bond. Again, that branch, that chain will be formed. So here again, it will be 1,4. So the point from where a branch is arising, that point will make a 1,6 glycosidic bond, and the inside the chain, the formation of a chain will have the 1,4 glycosidic bonds. So in this way, two types of bonds formation takes place to form one amylopectin molecule. So this is the in which amylose type of sequence is there, but this is the extra sequence which make it a branched structure. So starch has amylose and amylopectin. So here you can see, if we compare both of these structure of both of these types of starch, you can see a long chain will curl up and make an helical. But this amylopectin, you can see, so this, as for example, if we say this is the long chain, this is the long chain, so all they will have the 1,4 structure, and this chain from here, when these two molecules will join, a branch will arise. From that point, there will be the formation of 1,6 glycosidic bond. So in this way, we can differentiate between two types of forms of starch molecule. So here we are going on the next slide. The mixtures of amylose and amylopectin molecule build up into relatively large starch grains. You know, starch is a storing material. So it is stored in the grains, so which are commonly found in chloroplasts and in storage organs such as potato tubers and the seeds of cereals and legumes. You know, all they contain starch, which is stored in them. So starch grains are, if we want to see them easily with the light microscope, then we can rub a freshly cut potato tuber on a glass slide and stain it with the iodine potassium iodide solution. Iodine solution, simply we say which is actually the potassium iodide solution. So we can drop iodine solution over the slide, then we can see if the color is changed. That will be blue black from yellow or brown. So it means this potato tuber contains starch molecule. So this is the way the carbohydrates are stored in the plants. So starch is only a storing material into the plants. And the second type of polysaccharide which exists in animals, that is glycogen. You know, when extra glucose is there in the body, it is stored in the form of glycogen. In plants, the extra glucose is stored in the form of starch, but in animals, the extra glucose is stored in the form of glycogen. So glycogen is a multi-branched polysaccharide. Starch has branch structure, as we say that amylopectin, but glycogen is more branched than starch. So it is multi-branched polysaccharide of glucose that serve as a form of energy storage in animals, fungi and bacteria. So the polysaccharide structure represents the main storage form of glucose in the body. Both of they are the, you can say, energy storage, but they are present in animals and in plants. Starch in plants and glycogen in animals. Both of they provide energy to them. So if we discuss the structure of the glycogen, you can see over here, there is formation of 1,4 glycosidic bond and 1,6 glycosidic linkage. Both of they exist in them. That is glucose and glucose. They will join with this 1,4 glycosidic bond and then 1,6 glycosidic bond. So both of they exist in starch. Both types of bonds in glucose and starch, in starch and glycogen. So which are formed between glucose molecules. So the unit of both of them is the glucose molecule. So the glucose molecules will join together by two types of linkages, that is 1,4 and 1,6. So formation of glycosidic bond, and there will be the formation of starch and glycogen. But glycogen is more branched as comparison to amylopectin or starch. So after every 8 to 10 glucose residues, there is a branch containing 1,6 glycosidic linkage. If we compare them, both of they are the provides the energy, starch to plants, glycogen to animals. Secondly, both of they have 1,4 glycosidic linkage and 1,6 glycosidic linkage. But starch is less branched as comparison to glycogen. Glycogen is more branched as comparison to starch. So in this way, we can differentiate between them. So here in the next slide, you can see. So glycogen, the glucose storage polymer. You can see this is the structure of glycogen. Is the glucose storage polymer in animals is similar in structure to amylopectin. Already told you, it is similar in structure to amylopectin. That is, there is presence of both type of linkages, that is 1,4 and 1,6, but the highly branched structure permits rapid release of glucose. But this is highly branched, and these bonds formation is broken down as soon energy is required. So in muscles during exercise also stored in the liver, the ability to rapidly mobilize glucose to more essential to animals than to plants. So in this way, it is easily released when there is deficiency of glucose in the body of the animals. At that time, the linkage between the glucose molecules is broken down and glycogen is broken down into glucose molecules. So by releasing energy. So in this way, both of these storage organs are present. So another type of polysaccharide, the next subtopic is cellulose, which is another type of polysaccharide which is only present in the plants. So in plants, cellulose is composed and that gives rigidity to the cells because plants has to maintain their shape. So cellulose is present in the plant cell wall, which helps to maintain the shape, which protects the cell wall, which protects the plant cell. So plant cells are made of cellulose. The rigid structure of cellulose is what allows water to stand upright. If cellulose is not there, plants can fall down on ground. It cannot stand upright. So the bonds between each cellulose molecules are very strong, which makes cellulose very hard to break down. A polysaccharide consisting of linear chain of several hundred to many thousands of beta-linked D-glucose units. When we discussed glycogen and starch, so in that case, the alpha glucose was involved, but in cellulose, the formation of cellulose, the beta glucose is involved. But again, it is a long chain of beta glucose. So again, there is formation of glycosidic bond in the beta glucose molecules. But here, when there is 1,4 linkage, that is 1,4 linkage in this way, you can see. And the second, now this is the 1-glucose molecule, and this is the hydroxyl group in the upward direction. Hydroxyl group is in the upward direction. So the formation of glycosidic bond, one molecule has to revolve, that is both hydroxyl groups should be in straight line for the formation of a 1,4 glycosidic bond. So one projects above the ring in order to form a glycosidic bond with carbon atom where the hydroxyl group is below the ring. One glucose molecule must be upside down, or it has to revolve through 180 degree. It has to rotate or revolve around 180 degree for the formation of this glycosidic bond, as I will show you in the next slide. You can see over here. You can see that in the hydroxyl group in the beta glucose molecule, it is in the upward direction, but carbon number 1, but it only carbon number 1, the hydroxyl group of carbon number 1. So when there is the formation of 1,4 glycosidic bond, so one glucose molecule has to rotate through 180 degree. Through 180 degree, and there will be the formation of 1,4 glycosidic bond and the formation by the condensation reaction. Already told you what is condensation reaction, in which on joining of one glycosidic molecule between two glucose molecules, one water molecule is removed. So as a result, there will be the formation of glycosidic bond. So in this way, the formation of glycosidic bond will take place in the cellulose molecules, in which glucose molecule has to revolve through 180 degree. So you can see this is the structure of cellulose molecule, in which 1,4 glycosidic bond has been formed. You can see in this. So it will form in this way because the molecule has been revolved or rotated through 180 degree, but a long chain of cellulose is formed in this way. So it gives a great advantage. So this type of glycosidic bond give a great advantage to the plants. So cellulose derived from D-glucose, so which condense through 1,4-glycosidic. This is linkage motif contrast with that of 1,4-glycosidic. Already explained all this. So cellulose is a straight chain polymer. No coiling of branching occurs and the molecule adopt an extended and rather stiff rod-like conformation. So this is the stiff, stiffness is there in this structure. So we say the straight chains of cellulose can pack tightly together in parallel bundles. So in this way, they will tightly together in this way. So numerous hydrogen bonds between the hydroxyl groups are adjacent chains. So this extensive network of hydrogen bonds is what gives cellulose its high tensile strength. You can say, so when they make, so in this way, a crisscross arrangement formed, and so it gives a tensile strength and makes it insoluble and rigid fiber. Insoluble fiber, rigid. It gives rigidity to it. So in this way, uh, we can say that the structure of cellulose is defined by its long unbranched chains of beta glucose monomers linked by 1,4-glycosidic bond. So this unique arrangement allows for the formation of strong hydrogen bonded microfibrils. So these multiple hydroxyl groups on the glucose form one chain, form hydrogen bonds to same or neighbor chains, holding the chains firmly together. So in this way, you can see these are also chained together. So they also chain together. So in this way, when they make a chain together, then it will give a high tensile strength, which cannot be easily broken down. So this confers tensile strength in cell walls where cellulose microfibrils are meshed into a polysaccharide matrix. The high tensile strength of plants stems of the true wood also rise from the arrangement of cellulose fibers intimately distributed into the lignin matrix as well. So in this way, the cellulose molecule is highly rigid and flexible, which give a proper shape to the plant cell, and also it protects it to maintain its shape and to, you can say, keep the plant in the upside direction. So in this way, this is the very important type of polysaccharide molecule. So it was all about today's lesson. Please don't forget to like, share, and subscribe my channel, because this is the only way by which you can get the notification for my upcoming videos about past papers and syllabus of your class. Thanks for watching.